Every living organism on Earth depends on DNA, or it's called deoxyribonucleic acid. Your body, your brain, every cell inside of you. DNA stores the instructions for life, but we encounter a problem. DNA is too complex to have been the first biological molecules. It cannot copy itself alone. It cannot function without proteins, and proteins themselves require DNA instructions to exist. So scientists faced a paradox. Which came first, DNA or proteins? For decades, this question seemed impossible until researchers discovered something extraordinary: a molecule capable of acting like both, a molecule that may have existed before modern biology itself. RNA. What is RNA? RNA is a ribonucleic acid. It's often described as a middleman of biology. In modern cells, DNA stores information, while RNA carries instructions, and proteins do the most of the chemical work. For years, scientists assumed that RNA was only a messenger, but in 1980s everything changed. Researchers discovered that certain RNA molecules could catalyze chemical reactions on their own. These molecules became known as ribozymes. This was revolutionary because until then, scientists believed only proteins could function as enzymes. But RNA was different. RNA could store genetic information, fold into complex structures, and perform chemistry. In other words, RNA could potentially do the jobs of both DNA and proteins at the same time. This discovery led to one of the most important hypotheses in the modern origin of life science, the RNA World Hypothesis. The idea that before DNA and proteins, life may have been dominated entirely by RNA-based systems. According to the RNA World model, the first biological systems on Earth were not cells like bacteria. There were populations of self-replicating RNA molecules. These molecules may have competed, mutated, and evolved long before true life existed. This is one of the most fascinating ideas in science. Evolution may have started before biology itself. Before cells, they may have been molecular evolution. Others became more stable. Others folded into structures capable of catalyzing useful reactions. Natural selection could then act on these differences. The most efficient RNA systems survived longer and the less stable systems disappeared. This means that Darwinian evolution may have begun at the molecular level, not with animals, not with cells, but with chemistry itself. This is one of the most biggest scientific challenges that we face. Can RNA actually replicate? According to modern RNA, it's not fully self-replicating. Scientists still have not created a completely autonomous RNA system capable of indefinite self-replication under natural conditions. but research over the last decades has shown remarkable progress. Laboratory experiments have produced ribozymes capable of copying fragments of RNA. Other studies demonstrated that RNA molecules evolving improved functions over time through artificial selection. Researchers have even observed RNA networks cooperating chemically in ways that resemble primitive ecosystems. This is important because it suggests that the transition from chemistry to evolution may not require modern cells at all. It only requires three things: first, variation, replication, and selection. Once those existed, evolution begins automatically. One of the strongest pieces of evidence supporting the RNA world is that hidden inside of every living cell today, the ribosome. The ribosome is the molecular machine that is responsible for building proteins. For decades, scientists assumed that proteins powered this process, but detailed molecular studies revealed something astonishing. The catalytic core of ribosome is actually an RNA, not a protein. This means one of the most ancient and essential systems in biology still relies fundamentally on RNA chemistry itself. It is like a molecular fossil from an earlier world before us, a relic of the RNA era preserved inside every organism that is alive today. Even ATP, the universal energy currency of cells, and many important coenzymes contain nucleotide-like structures connected to RNA chemistry. The deeper scientists study biology, the more traces they find of RNA's ancient dominance. Despite its incredible abilities, RNA has major weaknesses. It is unstable. It breaks apart relatively easily. It is sensitive to heat, radiation, and chemical degradation. This creates a major question. How could this fragile RNA survive on the early Earth long enough to evolve? Modern research suggests that several possible solutions. Mineral surfaces such as clay may have protected RNA molecules and helped organize them. Certain metal ions may have stabilized RNA foldings. Icy environments may have slowed down degradation, and lipid vesicles, primitive membrane bubbles, could have stored and trapped RNA inside protected compartments. These structures are called protocells, not true living cells, but chemistry enclosed within boundaries. Inside protocells, RNA molecules could concentrate, interact more efficiently, and potentially evolve even more to more complex behaviors. This may have been the bridge between chemistry and biology itself. It is interesting that not all scientists fully agree on this classical model of RNA world. Some researchers argue that simpler self-organizing chemical networks may have existed before RNA. Others suggest that early life used alternative genetic molecules before RNA evolved. There are even hypotheses involving DNA, TNA, and other RNA-like polymers. Modern origin of life science is increasingly moving away from single-step explanation. Instead, many researchers now believe life emerged gradually through overlapping systems of chemistry, metabolism, compartmentalization, and replication. RNA may not have appeared instantly. It may have emerged from an evolving chemical world already increasing in the complexity. Still, RNA remains one of the strongest candidates for the first true genetic systems ever discovered. Eventually, evolution produced something more stable than RNA, which is DNA. DNA became the long-term storage molecule because it's chemically more stable and better for saving information. Proteins became specialized molecular machines capable of far more complex chemistry, and RNA remained in between them, still essential, still ancient, and still central to life. Even today, your cells still depend on RNA every second. Messenger RNA carries genetic instructions. Transfer RNA helps us to assemble proteins, and ribosomal RNA powers ribosomes. In many ways, life never truly escaped the RNA world, it simply evolved beyond it. Somewhere, billions of years ago, long before animals, plants, or even cells, certain molecules began copying themselves. They competed, they changed, they evolved, and from the ancient molecular struggle, all biology emerged. Every ecosystem, every organism, every human thought possibly could track back to RNA, not just a molecule, but perhaps the first bridge between chemistry and life itself today.